Welcome students. This class is for 4th sem students. Plant tissue culture. Lecture 1. I have specially made this lecture and the examination point of view. If it is helpful for you, kindly subscribe my channel so that update of my classes will be notified to you. And this plant tissue culture, I have followed the syllabus which is given by KUHAS, Kerala Health Science University regulation. Plant tissue culture, they have given history development of plant tissue cultures, type of cultures, nutritional requirements, growth and their maintenance, application of plant tissue culture in pharmacognosy, and edible vaccine, how they produce by the latest biotechnology innovative programs. In history of plant tissue culture, we would like to remember Hamberlanth 1902 first envisioned or given a spark in tissue culture, the concept of plant tissue culture, when he cultured cell of various species on a medium containing glucose of peptone, glucose and peptone. This is the man Hamberlanth. He is an American botanist, Gottlieb Hamberlanth. He lived in for 90 years, 1854 to 1945. He died in the time of Second World War. The history or historical milestones in plant cell tissue culture techniques are many, but only few I have taken. I have given a note for the students. You should refer more and make yourself a notes. It will be helpful for you. These are some of the year and some of the scientists and some of the work which I have selected. In 1892, Kelleher, first attempt to isolate protoplast mechanically. In 1902, we have seen that Hamberlanth, first cultivation experiment with isolated plant cells, cell growth, but no cell division obtained in Tradescantia species. In 1904, Hanning established of embryo culture for the first time, Cochlearia raffinus. And in 1960, Bergman, cell clones obtained from single cultured cell plated in an agar medium. The species he has selected is Nicotiana fasciosus. And in 1965, Vassel and Hildebrandt, they regenerated regeneration of a plant from one single cell cultivated in an hanging drop method. The species which they have used is Nicotiana. In 1983, Mitsui Petrochemy, first industrial production of secondary plant products by suspension cultures. The plant or the species which you used is Lithospermum. And in 1986, Sundberg Glemelius, he contributed the result of somatic hybridization or hybrids in Brassicaceae. The plant species of Brassicaceae is taken as for his experiment. Definition of plant tissue culture. Plant tissue culture, the growth of plant cells outside an intact plant, is a technique essential in many areas of plant sciences. It relies on maintaining plant cell in aseptic condition on a suitable nutrient medium. The culture can be sustained as a mass of undifferentiated cells for an extended period of time or regenerated into whole plants. The plant tissue culture is an essential component of plant biotechnology. Plant tissue culture, more technically known as micropropagation, can be broadly defined as a collection of methods used to grow large number of plant cells in vitro in an aseptic and closely controlled environment. This technique is effective because almost all plant cells are totipotent, ready to make cell division and ready to make the new seedlings. Each cell possesses the genetic information and cellular machinery necessary to generate an entire organism. Micropropagation therefore can be used to produce a large number of plants that are genetically identical to a parent plant as well as to another. So some of the points which we have discussed, that is in definition, the points to be remembered by the students. First one is aseptic condition, suitable nutrient medium should be added, undifferentiated cells will develop and from which generate into an whole plant, and plant biotechnology is a branch specifically from this plant tissue culture, and you can see that micropropagation of developing new plants as the plant cells are totipotent, having totipotency, and they are genetically identical plants can be developed. So this is what the definition, the points should be remembered when you write a definition for plant tissue culture. And this is just a picture to show you how a seed is taken in a petri dish in vitro study and giving a agar medium, nutrient agar medium, and the seedlings are developed in the petri dish itself, shoot system and root system and developed, and it can be transferred to the pot or to the ground for the development. This is just for an example, but we are not doing this one. We are taking explant, taking the part of the plant, a small part of the plant in an aseptic manner, and made them to give an environment of nutritional supplements which will be helpful for them to grow in vitro conditions. And in vitro conditions needed what all the in vitro conditions needed in the lab when you want to grow the plant. Freedom from competition. What competition? When you give a nutritional agar medium or nutritional medium, automatically microorganisms and other organisms can also develop very easily in that nutrient supplements which you have supplied. So freedom from competition should be there. So it should be a sterile aseptic conditions. The other organisms should not develop, and nutrition and a proper hormonal supply should be there, and a controlled environment. These are the 3 points which is very important to know when you are performing a tissue culture. So, in vitro study, the need of plant tissue culture, what all the precautions needed. First one, we need in vitro, what is the in vitro study need? First one is freedom from competition. The freedom from competition, bacteria, fungi and other organism which can be resisted to some degree by a whole plant, can easily outcompete for an isolated fragment of tissue from the plant in the relatively nutrient rich environment of a culture flask. This is usually done by chemical surface sterilization of the explant. Explant means a part of the plant or a part of the tissue which is taken from the plant source in an aseptic manner, and it is developed in an, or it can be incubated or inoculated to the media, nutrient media, with the proper supplements of all hormonal supplements and other adjuvants for additives and adjuvants for development of the plant with an agent. So when you are taking an explant, first it is with an agent as a bleach at a concentration. The concentration is very important, and for a duration that will kill or remove pathogens without injuring the plant cell beyond recovery. So we should use very low concentration which is only destroy the pathogenic organisms which will disturb our development, which should not disturb the plant cell. The minimum, or the medium which you are used, and the flask should be very much protected, and they must be sterile. The next one is nutrient and proper hormone supply. Nutrients and proper hormone supplies are needed. When a small portion of the plant is isolated, it is no longer able to receive any nutrient from the plant because it is already isolated. So you have to supply hormones from the plant, will not get. So you have to supply. These must be provided to although growth in vitro. So it should be supplied. These are types of hormones which you are adding. The amount of hormone will vary according to the tissue culture experiments which is performed, and which is the outcome which you are expected by the researcher. A controlled environment plays a major role. A controlled environment, piece of plant or an explant will which will be slowly divide and grows into a colorless mass of cell or proliferatory growth if they are kept in a special condition. Initiate from the most appropriate plant tissue for the particular plant variety. Presence of high concentration of auxin and a cytokinin growth regulators in the growth media. A growth media containing organic and inorganic compounds to sustain the cell. Aseptic condition during culture to exclude competition from the microorganisms is avoided. Factors affecting plant tissue culture. In this, there are 8 factors, especially light, temperature, genetics, genotype, explants, and carbohydrate sources, medium composition, additives, and plant growth hormones used. So there are 8. I have given an assignment for the student in this slide. By seeing this slide, you have to, all the students are informed to submit an assignment to think about and to refer about how these 8 factors affecting the plant tissue culture. So you should see that, you should see the reason out and how positive and negative light is needed or light is not needed, or what is the condition of light is needed, what is the condition of temperature needed, what is the, it is whether how it is affecting the tissue culture. All these 8 factors should be explained clearly with reference and submit an assignment to my office. Type of plant tissue cultures, they used to ask many questions many times. Last time before to exam they have asked types of tissue cultures and the principle behind the tissue culture. That means types only. We can write types of tissue cultures. There are mainly there are 4 types we have made, like isolated plant organ cultures, callus culture, single cell culture, and protoplast culture. And when you see isolated plant culture, excised root tip culture, excised meristem culture, leaf primordial culture, anther culture, embryo culture, and when you come to callus culture and single cell culture, the single cell culture is prepared from callus culture. So they are linked actually. So it comes suspension culture in the form from the callus culture. There are 2, batch culture and continuous cultures. In batch cultures there are 4. One is slowly rotating cultures, shake cultures, spinning cultures, stirred cultures. When you come to continuous cultures, there are chemostats and turbidostats. And when we come to protoplast culture, it is needed to know about how the protoplast isolation is done from the combined or the group of cells. So when you see this picture is simply to show that from carrot how they are making and tissue culture in the in vitro study by taking an explant, a piece of carrot which can be made as an implant in an aseptic condition in nutritional media. It develops callus, and that callus can be even made into suspension culture, and then embryo develop, an embryo will be developed, and automatically the plantlet, a carrot plant can be developed in this form. This is just a picture to show you, to show that explant. Explant means the living tissue which is cut into a small part, maybe a stem, maybe an underground stem, maybe a leaf, maybe a root, as part of tissue, living tissue removed from the organism or a plant and placed in a medium with the tissue culture. That is called explant. So when you want to see more, even an explant of a shoot, when you take, the shoot will be growth in a medium, it can develop root growth. And even if you take from a node, a shoot node can be taken, nodal segment, and also it can shoot growth, and you can see that is root growth can also be observed. And you can take a small piece in an aseptic manner, leaf disc can also be, callus culture can be developed in the nutrition agar medium adding, and from that shoot growth and root growth can be observed. So this is how the tissue culture developed. A simple picture to just to explain you. And the wound callus culture. So now we are coming to the callus culture and cell suspension culture together to explain, in which wound callus culture is the first name was given. These 2 of them are interlinked, especially callus culture can be, is the only thing is to give the source for single cell culture. And when you see in 1939, Gatherest in Europe and Mr. White, that is White, W should be capital, in USA reported that the tissues of carrot root explant proliferated to form large masses of disorganized tissues known as wound callus. When incubated on a nutrient medium containing mineral salts, glucose, cysteine, thiamine, and auxin, indole acetic acid, the callus is referred to a mass of undifferentiated, unorganized, parenchymatous cells proliferated on the isolated when growth on sodium medium, solid medium, not sodium medium, on a solid nutrient medium. Every callus cell has all the genetic information of the explant which you have taken for initial development. Under suitable nutrient condition, they will give rise to a new plantlet, new plant. Generally, a small percentage of cell of an explant in physically contact with the nutrient medium contribute to the formation of cellular tissues. Once a callus has been established, it should be subdivided and subcultured at regular intervals. For routine maintenance, a piece of healthy or health tissue 5 to 10 millimeter in diameter or 20 to 100 milligram fresh weight should be subcultured every 28 days. Generally, medium containing high auxin level, indole acetic acid, will induce callus formation. Inclusion of cytokinin, the growth hormone, with auxin may develop beneficially promotion of cells. For callus initiation, young cells are more suitable than matured cells. Germination of sterilized seed under aseptic condition will yield root, shoot and leaf materials suitable for callus initiation. For callus initiation from more specialized explant like pollen or endosperm, the media requires special modification when you take pollen or endosperm. Measurement of growth of callus culture was given by Nikhil as expressed in callus growth in terms of growth value of ratio of final size and the initial size. He states that Cullen growth increment is equal to final size minus initial size divided by initial size into 100. This is the Cullen growth where increment of size of callus can be measured. When you see this picture, it's a callus development from a carrot. When you take a carrot, a piece of carrot in an aseptic manner, and it is sterilized with water, it is washed and added with sodium hypochlorite solution and then washed with sterilized water and then transferred to petri dish, you can see the structure of the explant which you have taken from the carrot. It contains cambium, xylem, phloem separation in the cortex region clearly, which can be visible, and then it can be from the petri dish in a aseptic manner. It has been removed, it has been added to the culture medium in a conical flask at 25 degrees for 6 to 8 weeks. After you can find a callus development as shown in the picture. So this is what one example of a callus development from the carrot. Now we are coming to the suspension culture which can be prepared from the callus which is obtained. Suspension culture are derived from the callus tissue and are usually maintained in a glass conical flask containing liquid medium. Cell suspension cultures are widely used for studying pathways of secondary metabolism, enzymes, induction and gene expression, degradation of xenobiotics, and as a source of enzyme purification. The lack of chlorophyll and a carotenoid pigment in suspension culture helps to a great benefit for isolation of secondary products. Suspension culture represent a convenient form for the maintenance of cell line, biotransformation of added compounds, isolation and the characterization of mutants. You see this picture how the cell suspension culture is made. Callus culture tissue in a liquid medium can be kept in the conical flask and added and kept in the shaker, and cell suspension is obtained, and immediately it can be filtered and again centrifuged and resuspend in a fresh medium as shown in the picture 3 times, and then a counting of the cell suspension can be done. The suspended cell can be done in hemocytometer, can be observed, and from that a single cell or a uniform cells can be taken to plotting the cell in a suspension and shaken again, and so that the cell can even develop into an aggregates, and then from that embryoid and the regeneration of the plant can be developed from a single cell. So from here you can see the suspension culture, which I want to say there are, which we have seen, there is one is batch culture, which is having 4 types, like slowly rotating culture, shake cultures, spin cultures and stirred cultures. In continuous cultures we should discuss about chemostats and turbidostats. So if you see that basically there are 2 types of suspension culture. One is batch culture, another is continuous culture. In batch culture we have 4, that means batch cultures are referred to be closed system. A fixed volume of medium which is agitated to maintain the oven distribution of cells to promote adequate gas exchange with the air. The 4 types of batch culture system are in current use, and they are distinguished by the way which movement of the liquid medium is achieved. Number 1, we have discussed slow rotating cultures, shake cultures, spinning cultures and stirred cultures. This is the slow rotating cultures. Single cell and the cell aggregates are grown in a special designed flask, the nipple flask. It has 8 nipple as shown in the picture. A nipple flask possesses 8 nipple like projections. The capacity of each flask is 250, and you can see the picture how the disc of 8 discs are there, and the picture they have shown 1, 2, 3, 4, 5, 6, 7, 8. So in that 8 disc, how the cell suspension nipple are rotating next. You can see the cotton plug in the middle. So this can give an, this is a batch type of slow rotating culture flask. Large disc rotated 1 to 2 revolution per minute can be used. This is the type of shake cultures where we can see the very simple and effective system of suspension culture. In this method, simple, not single cells and cell aggregates in fixed volume of liquid medium are placed in conical flask. The conical flasks are mounted with the help of clip on a horizontal large square plate of an orbital platform shaker. The square plate moves by a circular movement at the speed of 60 to 180 revolution per minute. You can see the flask in the picture and having a centric bearing of shaker. Speed control is also there. This is actually a spin culture, slanting of 45 degrees. A large volume of cell suspension may be cultured in 100 liters also. Bottle can be kept, which are rotated in a culture spinner at 120 revolution per minute at an angle of 45 degrees. You can see the picture. Upper bearing and lower bearing will be there in between the 10-liter bottle with a cotton plug and a cell suspension which is slanting with 45 degrees. of rigid frame as shown in the picture. This is actually the stirred culture. This system is also used for large scale batch culture in 1.5 to 10 liters can be used. In this method, the large culture vessel is not rotated, but the cell suspension inside, you can see there is a picture MS, that means the magnetic stirrer will be there. The suspension inside the vessel kept dispersed continuously by bubbling sterile air also through culture medium. Use of internal magnetic stirrer is most convenient way to agitate the culture medium safely. The magnetic stirrer revolves in 200 to 600 revolution per minute. The culture vessel is 5 to 10 liters round bottom flask as shown in the picture. And you can see the continuous culture system, we know there are two. This system is very similar to the stirred culture. Only thing is here it is continuous, replacing the old one medium with the new one. But in this system, the old liquid medium is continuously replaced by the fresh medium to stabilize the physiological states of the growing cells. So there are two, that is chemostats and turbidostats, which we have discussed previous slide itself. So now we will see the chemostats. This is a system of culture. Vessels are generally cylindrical and circular in shape and possess inlet and outlet pores for aeration. You know that it is a continuous process, and introducing of and removal of cells and medium. The liquid medium containing the cells is stirred by a magnetic stirrer. The introduction of fresh sterile medium which is pumped, there will be pump will be there for pumping in the constant rate into the vessel, balancing by the displacement of the equal volume of spent or old medium and the cell. Such a system can be maintained in a steady state so that the new cells are produced by division at a rate which compensates the number loss of overflow of spent medium. So this study or these type of thus in this steady state condition, the density, growth rate, chemical composition and the metabolic activity of the cell all remain constant. Such continuous cultures are ideal for studying growth kinetics and the regulation of metabolic activity in higher plants. So next, even the picture you can see chemostat of the continuous culture method or tissue culture method. In this cell suspension culture method, you can see the round bottom wide flask with a magnetic stirrer and probe for a sampling, and also pumps are there for inside pumping the new suspension, and also pumps are present to remove the solutions also. By suction can be used, and the aeration also done with sterile for circulation. Now turbidostats in the continuous process, you can see they are turbidostats. The turbidity of the suspension culture medium changes rapidly when the cells increase in number due to their steady state growth. So when the number of cells division is increased and cell increased, the population increase, automatically turbidity will increase, and the changes of turbidity of this culture medium can be measured by changes in the optical density by means of using a photocell or turbidometry of the medium. Again, the pH also will change if there is more populated cells, cell division is more and the density of the cells are increased. The pH of the medium changes due to the increase in cell density. In turbidostats, an automatic monitoring unit is connected with the culture vessel, and such unit adjusts the medium flow in such a way as to maintain the optical density or the pH at a chosen preset level. You can see the picture, that is photocell controller or pump can be also medium fed of the maintain the turbidity, and terebinthostat is having a stirrer also, and pedal stirrer, and you can see surface medium and also culture volume kept constant by overflow harvesting device. Light can be also used in this way, and also the photocell, the transmitted light can be calculated, and turbidity will give you the parameters of, or the rate of cell division can be identified. Now the last one, protoplast culture, which we have seen, that is isolation of protoplast from the culture is the main event in this type of culture. So protoplast culture, naked plant protoplast, that is removed cells. Naked plant means cells removing of the cells by means of using degradation enzymes, and the most common used enzymes are purified preparation of cellulase, hemicellulase, and pectinase obtained from fungi. Pectinase usually break down the pectin and holds cell together. Cellulase digests the cellulose in the plant cell wall. Isolated plant protoplast provide basic information for the genetic modification of the plant cell. Somatic hybridization, plant breeding, and in the study of virus infection. The isolated protoplast can be induced to fuse with protoplast and different species, that providing an opportunity for creating new plant hybrids. The isolated protoplast is an ideal recipe for foreign DNA, and transformation can lead to the production of genetically modified plants. So when you see how in the picture you can see that protoplast cell wall formation and first division takes place and four cell conditions, and then microcalli formation and macrocalli formation, and which can develop into roots and shoot system, and regeneration of the plant can happen. And in protoplast, mainly protoplast isolation, some of the points have to be remembered. The successful isolation of protoplast depends on many factors, such as source of the tissue which you are taking, what plant organ, embryo, cell suspension or callus. And plant species also have a factor influencing in isolation. Physiological status of the plant or a cell suspension, nature of the cell wall degradation, enzyme mixture, and the protoplast visibility, and the success of protoplast culture depend on their ability to regenerate new cell wall and recovery capacity for division and growth. Protoplast culture, this is the picture which you can see. There is a young plant. From that explant of tissue sterilized, of the leaf sterilization can be done, and the epidermal peeling will show you the peeling leaf segment which can be that is plasmolysed. Cells can be added with enzymes mixture like celluloses or peptilases, which can give the cell wall digestion and release the protoplast, and protoplast can be taken in the test tube as shown in the cell debris can be washed for protoplast, and isolated protoplast can be planted in protoplast cell suspension, and cell wall generation takes place, and clubbing of cells, callus tissue which can develop a or differentiation of the callus tissues can be developed, and which can plantlet derived from the protoplast can be developed in tissue culture. This is what the picture which shows how a protoplast culture is made. So we have seen almost all the definitions and different types of protoplast and what all the factors which is affecting. And I want you to, these 8 factors which is affecting the tissue culture, which is given an assignment, just write in detail and with reference submit to my office. Thank you. Thank you for being with a small class of introduction class, and with one more we have to continue this tissue culture in the next sector. Thank you.